This begins recorded lecture number 2 for students in Ms. Stout's biology class at Limestone Community High School on the unit of the plasma membrane and homeostasis. In the first unit, we talked about the general look of the plasma membrane. We talked about its function and a little bit about how each of those parts works. Now we're going to look more at the structure of the plasma membrane and the individual pieces itself. The plasma membrane is described as a bilayer, meaning it's made of 2 layers, and those 2 layers are actually made of phospholipids, and that's what they look like. Kind of looks like a perfectly round little balloon with 2 strings hanging down, and then on the other side is basically the same thing. So these are called phospholipids, and I encourage you to add that to your notes. Phospholipids. So each layer is a sheet of lipid molecules, and a lipid is just a fancy science nerd word for a type of organic compound or organic molecule known as a fat. So lipids mean fats. This is a way fancier illustration of basically the same thing. This is still a phospholipid. You can see that it has what is known as a hydrophilic head. Well, philic or phile means to like or love, so it's water-loving. And then the tails, you can see it says phobic, hydrophobic. Phobe refers to an aversion or fear of. So one part of it loves water and the other part of it doesn't love water, and we'll talk about how that's pretty significant with how the membrane is put together and how it works. Embedded all the way through the membrane are membrane proteins, and here again is another example of the fluid mosaic model. And again, I'm showing you several different ones because I don't want you to just focus on like one specific example, because what I want you to know is the overall concept. We have, again, the phospholipid bilayer, and then extending all the way through, these kind of look like slices of raisins in a raisin bread. We have the proteins that go all the way through. Now, another thing that you can see this in this example, which I do like, is that there are some cholesterols on each side of that lipid bilayer. And cholesterol is just another type of fat, and usually it gets a bad rap, but it is good to have, it's necessary actually, to have some cholesterols in your plasma membrane. Continuing with the structure. So those phospholipids are made up of 2 parts. You have the head, which is the the water-loving part, and you have the tail, which is the non-water-loving part, or the water-not-loving part. The tail is made of 2 fatty acids, and those are considered to be non-polar. If something is non-polar, what that means is there's no overall charge to the molecule. If something is polar, then it means there is an overall charge. And when I say charge, I mean there's a slightly positive end and a slightly negative end. So on your test, not only will you need to know the phospholipid, you'll need to know that the tail part does not like water. It is insoluble in water. It means it does not dissolve in water, and that makes sense if you think of it, because fats repel water. Oil repels water. They don't mix. And that is because of that non-polar structure. Water itself is a polar molecule. It does have a slightly positive and slightly negative end. And, excuse me, the head of the phospholipid is polar. It does like water, so it is soluble or dissolvable in water. Well, if you remember from when we talked about the water cycle, we said that living things are made of 70 to 90% water. Well, cells are living things too, and so cells are not only in a watery environment, but the inside of their cells are a watery environment too. So the phosphate end, the phosphate head, will align toward the water both inside the cell and outside of the cell. And so the 2 layers are kind of like a mirror image of each other. So in the inside of that watery environment, you're going to have the head of the phospholipid aligned that direction, and then also on the outside. And so those non-polar tails, those not water-loving tails, will be on the inside of the bilayer. Now what this image is showing you is kind of like a cross section of a cell. So if you just cut a chunk out, so you can see the watery environment outside of the cell, and then we'd have the cytoplasm inside of the cell, and you've got those phosphate group heads towards the watery environment, and then the non-water loving non-polar tails on the inside of the bilayer. More about phospholipids. The shape of those phospholipids, as you can see, the phosphate group head, it is round. And that's not just because that's what the illustrator wanted to make them look like. That's actually what the molecule looks like. It's round. So because of that, you can kind of imagine the classroom being filled with round helium balloons. And they would easily be able to bounce around within their own layer. That same exact thing happens in the fluid mosaic model. Those phospholipid molecules can move sideways through their own layer, but they will not flip from top to bottom. Another part of the fluid mosaic model is going to be those proteins. And the proteins are going to also be associated with specific organelles. That's why the organelles within the cells can do different jobs too, because they have different proteins. Proteins that help the Golgi body package up fats and lipids will be one shape, whereas proteins that help the mitochondria release energy from the food that you eat are going to be a different shape. This is kind of more of an electronic version of what the fluid mosaic model could look like, but again, same concept. We've got phospholipids on one side, inverse phospholipids on the other, and this is showing you two different types of membrane proteins. You can see that they extend all the way through the membrane, and they have different shapes because they would be associated with different molecules. Because of these different shaped proteins, that's how selective permeability works. Remember, selective permeability is the property of a membrane that allows some materials to pass through while keeping others out. Not only does that work with the whole cell, but that also works with the organelles within the cell. And the last thing we're going to do in this lecture is diagram that fluid mosaic model. So you do have this diagram in your notes. You're going to want to add these labels to the diagram. First, you have the phospholipid. Phospholipid are those round balloon-like structures that extend on both sides of the membrane. We have a cholesterol. The job of the cholesterol is to lend the membrane support and stability, because you want everything to be moving fluid but not too fluid. It keeps it so that it just doesn't fall apart. You have membrane proteins, and actually we have three different membrane proteins. This is another one and a third one. You can see that there are different shapes, obviously, because they would do different jobs. Just for illustrative purposes, we have the phospholipid on the other side of that membrane too, and then we would have the cytoskeleton to show you that this is actually the inside of the cell. Next would be diagramming the phospholipid itself. We know that it's a phospholipid not only because I'm telling you, but because it's going to have the two parts of the phospholipid. We have the phosphate group, a phosphate group, which phosphate can be abbreviated PO4. It is polar, which remember that means that it likes water, and it can be dissolved in water. The other part are the fatty acid tails, nonpolar, so does not love water, and water will not dissolve it. And that is the end of lecture 2 for the fluid mosaic model, homeostasis and the plasma membrane.